Good afternoon, everyone. My name is Lauren Russell and I work in the Centre for Food Safety in UCD with Professor Seamus Fanning. And today I'm going to give you a little overview about the identification and prediction of Listeria biocide tolerance in dairy processing. So a little bit of an overview of my talk today. I'm going to tell you about me, a little bit about me and who I am, why you should maybe listen to me. What is Listeria? I know we have a very diverse audience, so I wanted to make this kind of non-technical so that everyone can get something from this. The implications of Listeria in dairy processing. What are biocides? How are they used in the dairy industry? And what we know about biocide resistance. A little bit of the basics of whole genome sequencing, although I think Shay has told you more than enough about whole genome sequencing. A little bit about minimum inhibitory concentrations and how we use them to determine biocide resistance in Listeria. And finally, how we can use whole genome sequencing to identify and predict Listeria biocide tolerance. So a little bit about me. At the minute, I am a molecular microbiologist at the UCD Centre for Food Safety in Dublin. I graduated this year, as Francois said, with my PhD in microbiology from UCD, which was funded by the Department of Agriculture in Ireland. The project investigated the public and animal health risks associated with land spread of anaerobic digestate and slurry on land in an effort to add value to the AD industry and to inform policy. Since January of 2021, I've been working in UCD CFS, and I started with the SARS Food Project, a piece of work funded by WHO to assess the risk of COVID-19 in the food chain. So as part of that project, we sampled a range of foods from retail outlets in Ireland and tested the foods themselves and the packaging for the presence of SARS-CoV-2. At that time, I was also part of the Irish COVID study group, so we were carrying out whole genome sequencing to identify variants of concern in Ireland. And most recently, since June of 2021, I've been working on a biocide tolerance project with bioMurio to predict biocide tolerance within not just the dairy industry, but the meat industry as well, and that's what I'm going to talk to you about today. So first of all, what is Listeria? Listeria is a non-spore forming rod-shaped bacterium which is often present in the food chain. It's the causative agent of listeriosis, which can have serious implication for vulnerable individuals, such as the immunocompromised, pregnant women, the elderly, and young children. So of all zoonoses under EU surveillance, listeriosis has the highest rate of hospitalisation and thus is a major public health concern. In normal, healthy individuals, infection can be asymptomatic or may cause fever, muscle aches, nausea and diarrhoea. However, in predisposed individuals, symptoms can be far more severe, often resulting in hospitalisation and death, and can cause miscarriage and stillbirth in pregnant women and sepsis and meningitis in the elderly or children. So the implications of Listeria in dairy processing. Well, milk and milk products are frequently identified as vectors of transmission for Listeria. Milk can be contaminated at farm level, either by indirect or external contamination from the farm environment, or less frequently by direct contamination of the milk from infection in the animal. Pasteurisation of milk will kill Listeria, but post-pasteurisation contamination, as well as consumption of unpasteurised milk and milk products, can lead to milk being the cause of a Listeria outbreak. Therefore, it is a concern that Listeria in milk could lead to a public health risk. In order to protect against this risk, there is a need for awareness surrounding the issues, hygienic practices to reduce the incidence of Listeria and risk analysis. So Listeria has long been reported to be more thermotolerant than other pathogens, with studies reporting survival at temperatures between minus 0.4 and 45 degrees Celsius, making Listeria a psychrophile. Listeria have been documented to be resistant to a wide range of stresses, including low pH, high salt concentrations, which allows them to survive better than other foodborne organisms. Listeria possess the ability to form biofilm. Sorry, Siri is talking to me.
I didn't get that. Could you try again?
I don't know how to turn her off.
It's not a problem.
Oh, sorry. Listeria possess the ability to form biofilm. For those who might not be microbiologists, biofilm is basically when bacteria attach to the surface and they grow in sessile communities. They form an extracellular matrix or a mucus-like layer which gives the bacteria more protection from the environment, allows it to avoid desiccation, nutrient deprivation or disinfection treatment. And certain Listeria strains are stronger biofilm formers than others, which allows them to persist longer in the environment. And finally, Listeria may be resistant to biocides, which is what we're going to talk about. So what are biocides? Under EU legislation, a biocide is a chemical substance or microorganism intended to destroy, deter, render harmless, or exert a controlling effect on any harmful organism. They are commonly used to counteract bacterial contaminations in a range of applications, mostly as infection control and hygiene measures in hospitals and in food production. Biocidal products contain one or more biocidal active substances, but may also contain other non-biocidal co-formulants which may aid in effectiveness or deliver desired qualities, for example, pH, viscosity, odour, colour, etc. All biocides must be registered under legislation, that is the Biocidal Products Directive in the EU and the Federal Pesticides Law in the USA. So how are biocides used in dairy processing? So as part of the project, I carried out a survey of biocide use in the Irish dairy industry through contact with plant operators and chemical suppliers. The survey indicated that biocides are used in the majority of cleaning processes, including CIP and COP cleaning, automated and manual processes, as well as floor cleaning, heavy-duty cleaning processes, surface disinfecting and hand sanitising. The most common biocidal ingredients in the dairy industry were hydrogen peroxide, sodium hydroxide, sodium hypochlorite, ethanol, isopropanol and peracetic acid. While benzalkonium chloride was mentioned in one or two products, it's clear that quoc usage is being minimised. An international survey was also carried out to capture biocide use in the UK, EU, USA and China. The international survey showed similar results, however, quoc usage was more common outside the EU, while within Europe a move away from quocs is clear due to current regulations surrounding quoc residues. The other difference was that Chinese markets reported the use of biocides for other uses than cleaning, namely carcass decontamination of poultry and shelf life extension in fruit and vegetables, so not specifically within the dairy industry. What do we know about biocide resistance? Well, unlike antimicrobial compounds, which are selectively toxic, most biocides do not have distinct bacterial cell targets on which to act. Antimicrobial resistance normally arises following one or more target gene mutations. However, biocide resistance is mediated by mechanisms which are less well characterised. These mechanisms can include the upregulation of efflux pump activity and structural alterations to the cell wall, which can impact permeability. In the modern food industry, the scale of food production due to consumer demand for healthy nutritious and minimally processed food devoid of additives, such as chemical preservatives and other antimicrobials, has had an impact on the volume of biocides used in the industry. And in an attempt to improve hygiene measures and ensure food safety, the industry has increased use of biocides and chemical-based disinfectants to control the microbial ecology of the production environment. This increased use has led to reports of biocide tolerance for most classes of agents, which has important implications for public health. The development of biocide tolerance could compromise the role of disinfectants as an effective means to control bacteria, and any failure of these disinfection protocols due to biocide tolerance could facilitate the survival of pathogenic organisms and contribute to the emergence of persistent strains. So, a little bit about whole genome sequencing. As we all know, organisms have a unique genetic code or genome. This genome contains a range of nucleotide bases, A, T, C, and G, in a very particular order, and this order dictates the genes that are present in the organism. Whole genome sequencing is a process where you can determine the sequence of these bases and identify its unique DNA fingerprint, and you can use techniques like SNP analysis. Whole genome sequencing provides an abundance of information which can provide insights into the behaviours and characteristics of an organism, for example, the antimicrobial resistance profile, and can be used in outbreak tracking. So what is SNP analysis? SNPs are single nucleotide polymorphisms. There are single nucleotide changes in the genome, and analysis of these SNPs can show subtle differences between strains on a more specific level, from the full gene differences right down to single nucleotide. SNP analysis has a range of applications, for example, outbreak tracking, where these small similarities can be used to connect strains of the same outbreak. The SNPs occur during cell duplication and allow tracking of bacterial change from one generation to another, and in this way have also been used to determine changes in bacterial lysis pre and post exposure to compounds. The genome sequences can be mapped to reference sequences and compare the SNP differences. So what is minimum inhibitory concentration? I always struggle with that one. So the MIC is defined as the lowest concentration of a substance that inhibits the visible growth of a given bacterial strain. MIC determination is important in clinical settings to ensure that antibiotics are chosen efficiently to increase the success of treatment. They are one of the primary tools for assessing resistance levels and monitoring changes in resistance. And MIC methods can vary from agar dilution to broth, micro and macro dilution methods, and this can lead to vastly different results, so it can be an issue sometimes. The minimum bactericidal concentration is the minimum concentration of a substance which kills 99.9% of a test organism. MBC determination is important in the context of resistance to ensure complete destruction of organisms exposed to active substances and reduce the risk of acquired resistance. So how do we determine biocide tolerance in Listeria? The aim of the study was to generate MIC and MBC measurements in the lab in combination with whole genome sequence information to identify and predict Listeria monocytogenes resistance to biocides of interest to the dairy industry. Benzalkonium chloride was selected as a long-standing biocide of use in the industry, and because targets for BC resistance in Listeria have been reported in the literature, for example, the BCR ABC cassette. Peracetic acid is an emerging biocide which has become increasingly popular in place of BC, and targets for peracetic acid are not known at this time. Listeria monocytogenes was selected for the study because of its prevalence in the food chain, nature as a persister, and because its modes of resistance are widely reported. 160 strains were selected from the UCD CFS strain bank. For all of these, the whole genome sequences are available. A literature search was carried out to identify potential genes of interest in each target organism, genes related to specific resistance mechanisms for the actives chosen, as well as non-specific targets, genes related to biofilm formation, efflux pumps, and general persistence were considered and used in the strain selection process. These factors were used to curate a strain collection which was diverse and represented both presence and absence of these genes in a way that was most likely to give us an answer. So, as I mentioned, there can be issues with MIC determination, and we wanted to use a standardised method for MIC evaluation. So for this study, we used this method that was developed in 2020 by Shug et al. It's a microbroth dilution method which is standardised to allow consistent, comparable results, and uses less materials, allowing for a higher throughput. Basically, I won't go into too much detail, but the protocol involves growing up your bacteria, preparing McFarland standard, which ensures that your strains begin at a similar concentration. You add distilled water to each well, you add your active ingredient to your first column, and you dilute so that you get a gradient across the plate, and then you dose with your bacterial suspension. So then you incubate your strains overnight, and you'll be able to visually see the following day. I'll show you a picture in a minute, which is the MIC. So in order to try and increase the throughput and increase the reliability of results, this MIC plate preparation step was automated using a liquid handling system. So, little video of my robot. He's a lot slower than me, but he frees up my time, so it's okay. So, here's an example of some of the MIC plate results. On the left-hand side, you will see the benzalkonium chloride test plate, and on the right-hand side, peracetic acid. You'll clearly see on the right-hand side of each plate the turbidity of the wells, which is the growth, and you can clearly see where that growth cuts off. And the MIC is marked as the first column without visible growth. These plates are then used to carry on to determine the MBC values. So as previously mentioned, the MBC is the minimum concentration of a substance which kills 99.9% of the organism. The method for the MBC is extremely simple and follows on from the MIC plate, where a loop full of the MIC and 2 dilutions above are plated out on nutrient taker and incubated at 37 degrees overnight. So following incubation, the plate is examined and the MBC is assessed visually again, and the first dilution at which the strain is eliminated is your MBC value. So how does this all tie together, and how can we use MIC and MBC data with whole genome sequencing to identify and predict listeria biocide tolerance? So to do this, you use a method called genome-wide association study, or GWAS, with your SNP analysis. It's a research approach used to identify genomic variants that are statistically associated with a particular trait. The method involves surveying the genomes of many strains, looking for genomic variants that occur more frequently in those with a specific trait or gene to those without the gene. So you basically compare your phenotype and your genotype to try and draw links between our genes and what you're seeing. So just to show you a few results, I just want to give you some information on clonal complexes. I don't want to categorize things and confuse people. So listeria can be, or bacteria in general, can be categorized into groups called clonal complexes. Each organism possesses 7 housekeeping genes, which are cellular maintenance genes, which regulate basic and ubiquitous cellular functions. And to be classified as a clonal complex, strains must possess at least 5 similar housekeeping genes in common with each strain of the clonal complex. So, the reason I said was because of this. So, this slide shows a summary of the MIC and MBC results by clonal complex. So, clonal complex 1 is generally associated with infection. Clonal complex 2 usually harbours benzalkonium chloride resistance encoding genes, which are associated with and are associated with infection to a lesser extent than clonal complex 1. However, the presence of the benzalkonium chloride resistance is not really reflected in the results that we saw. However, clonal complex 5 exhibited high MIC values, likely due to the presence of efflux genes and benzalkonium chloride resistance cassette. CC5 isolates are isolated from environmental swabs in the production environment. Clonal complex 8 was similarly associated from environmental swabs, but does not possess benzalkonium chloride resistance. This likely explains the lower MIC and MBC values that you'll see there. Clonal complex 9 and CC121 are commonly related to food. Similarly, certain CC9 isolates were BCR ABC positive. All strains were positive for efflux genes, as well as hydrogen peroxide resistance, and 5 genes involved in stress survival Ireland 1 were present, which allows the strain to survive under stress. CC288 is isolated from the production environment as well and possesses a range of interesting genes, including those encoding efflux pump genes, the LIPI3 island, which is linked to hypervirulence, as well as hydrogen peroxide resistance genes, and the BCR ABC cassette, which, as I mentioned, confers benzalkonium chloride resistance, which explains the high MIC and MBC values. Peracetic acid has proven to be extremely effective biocide, with very low MIC and MBC values obtained in all strains. And in this way, further tests are required in order to try and pinpoint the genes responsible for tolerance in Listeria. This maybe might come through, we're doing some other work with other species, so it might shed some light on some targets for peracetic acid. So how can we apply this for the dairy industry? Whole genome sequencing allows for information-led, precision-based approach to food safety and will change the way that we approach risk assessment. Sequencing of outbreak samples gives us an insight into possible resistance, allowing changes in cleaning processes to avoid emergence of persistent strains and reduce time spent managing outbreaks. SNP analysis allows us to track changes in the bacterial genome and may in future answer the question of how pathogens colonise the production environment, making whole genome sequencing a very powerful tool for food safety. And I would like to thank Shay and all of the team in UCD, as well as Joao, Kathleen, Dieter, Xavier and David from BioMario for all your help. Thank you.